I'm Christine Vokey, Director of Education and Information at PXE International, and we are very pleased tonight to have Dr. Jouni Uitto with us to tell us about mice and PXE. The title of his presentation is, Do Mice Have PXE, and Why Study Them? Dr. Uitto has been looking at PXE for a long, long time, and I think one of his big claims to fame in our world anyway is that he has developed a mouse with PXE, and I hope he'll explain that to us. He's at the Department of Dermatology and Cutaneous Biology at Jefferson Medical College at Thomas Jefferson University in Philadelphia. And I think with that, I will turn it over to you, Dr. Uitto.
Okay, thank you, Chris. Good evening, everybody. Greetings from Philadelphia, the city of brotherly love, as they say here. And yes, as Chris said, I've been working on PXE for a long time. In fact, my first publication on PXE was in 1971, and we are still very actively working on this condition, and progress is being made. So the topic tonight is really the question: do mice have PXE, and if so, why study them? But just before going to the mouse studies, let me just remind you of a couple of features of PXE, which really then allow us to compare the human situation with mice that have PXE, or at least very much PXE-like condition. So first of all, the PXE, as you certainly may know, it's for many of the general practitioners, physicians, it's a diagnostic challenge for a number of reasons. One reason is that it's really a group of diseases. In other words, not every PXE looks the same, and there is quite a lot of variability, so that the spectrum of clinical manifestations is highly variable. Some have milder disease, some have more severe disease, and there's considerable clinical heterogeneity, which means that some people have problems mostly with the skin. Others may have mostly eye problems, and in some cases the vascular problems are the ones causing most trouble. And of course, a challenge to the diagnosis that the onset of manifestations is late, so it's not present in most cases at birth, but usually in teens or even 20s, 30s, the diagnosis is being made. So it's a multi-system disorder affecting skin, eyes, and the blood vessels in the cardiovascular system. And as I said, in some families, one of these organs can predominate, so that the skin is major problem, eyes or cardiovascular, etc. I'm sure most of you are familiar with the typical skin findings, which often lead to diagnosis. This kind of yellowish little papules, a typical place on the arms or at the side of the neck. And with time, the affected individuals get more of these papules, and they tend to coalesce into this kind of inelastic, almost like leathery type skin, and can affect most of the skin with time. Of course, this is mostly a cosmetic problem, but can certainly cause major psychological and cosmetic issues with the patients. Now, if you take a little piece of skin from affected area, you find that there is this kind of unusual dark structures. These are so-called elastic fibers in the middle of the skin. Here is the outside of the world, top of the skin. This is the middle part of the skin, and there is an unusual amount of these elastic fiber structures. Now what is unusual in these, besides they look a little thicker than the normal elastic fibers in this part of the skin, but they also become calcified. In other words, calcium phosphate complexes deposit on these elastic structures, and that causes the inelasticity, kind of leathery texture to the skin. Now, of course, the eye problems are the major ones causing a significant amount of problems. Again, the pathology in the eye, so-called angioid streaks, that is caused by breakage in the elastin-rich layer in the back of the eye. And that is due to, again, calcification of these structures. And this causes breakages. And when they break, there are blood vessels which grow, but they are very fragile. They break and they leak blood to the tissue, and that results in the loss of visual acuity. In other words, the vision gets worse, especially following trauma. So that's something people certainly should avoid. And this just shows the bottom of the eye. There are the little breaks. These big ones are blood vessels, and then there are breaks which cause the breakage of these bigger blood vessels and cause the bleeding. And then finally there are the cardiovascular problems. The blood vessels become calcified. They can break. They can result pain in the lower legs when you walk a little fast and longer periods, and occasionally can cause coronary artery disease at the earlier than usual age. So over the years, we were studying the cause of PXE and didn't have a very good idea until in late 90s when PXE International organized a consortium of 4 scientific groups. And these groups went by systematically looking into the human chromosomes and human genes, and they ended up with this so-called ABCC6 gene as the site where the mutations in a PXE reside. And this ABCC6 actually encodes, in other words, it has the blueprint for a protein, so-called ABCC6 protein, which is a transmembrane pump. Basically, it pumps something out of the cells to the blood circulation in normal situations. And we can actually find mutations by this kind of they look like complex machines, but they actually are pretty straightforward once you get used to, and you have these well-trained biomedical scientists. This is actually my picture when I used to have a beard. That's probably 10 years ago or so. And what it does, it finds mutations, and for example here is the ABCC6 gene, and all these arrows point to positions of different mutations in that gene. As you can see, there are lots of mutations, so most families, they have their own private mutation, which however causes this protein to be non-functional. You see here is more mutations. They concentrate on certain critical areas of the protein, but the end result of all these mutations is that the pump does not work. Whatever it pumps from the cells to the outside to the circulation, that does not happen in case of PXE. Now, one of the puzzling observations early on when we realized that the ABCC6 gene harbors the mutations in PXE was that it's expressed primarily in the liver. It's not expressed in the skin or the eyes or the blood vessels. So really the question is, how do mutations in this gene then lead to mineralization or calcification of the connective tissues in the skin or eyes or the blood vessels. And that's a real challenge. One question was really, is PXE primarily a metabolic disorder? In other words, the ABCC6 gene is primarily expressed in the liver. So as a secondary one, we get some changes in the blood, in the circulation, and these molecules, or lack of them in the circulation, then causes the mineralization. That's the question. And one approach to look into this is develop PXE mice. In other words, what you do, you mimic the mutation. Basically, you eliminate this gene in mice by techniques. They are rather complex techniques, but they are in routine use. And so you can eliminate this gene and see if the mice now develop PXE. And this is just shows some of the complexities of the technology, but basically you human or mouse ABCC6 gene has a total of 31 different coding segments, and you eliminate just in the middle of them from exon 11 to exon 19, you eliminate piece of the gene, and then the gene does not make a functional protein, so therefore the ABCC6 is not functional. And this just shows that we have knockout mice. Here there's no wild-type normal allele here at all. And we can show in the liver, this is normal mouse. You see this green color? That's where the ABCC6 protein is in the liver. Just on the surface of the liver cells surrounding the cell, if we look at these mice where we have deleted the gene, there's clearly no protein being made at all. So these are so-called knockout mice, or ABCC6 null mice, or minus mice, so they don't have this particular protein in their gene. So we looked into those mice, and when they were born, they looked perfectly normal. You look a month, they are perfectly normal. They look 3 months, they look perfectly normal. But when we started biopsying, taking skin biopsy, we noted that these mice have mineralization in their vibrissae. Now, vibrissae are the whiskers of the mice here, and the whiskers have a big hair, and this is a histology of the whiskers. Here is a hair. It's surrounded by the sinus. It's a blood vessel, and surrounding the blood vessel is this kind of connective tissue capsule. And if you now stain with special stains, this capsule becomes mineralized, like the tissues in human patients with PXE. And in fact, this mineralization can be noted as early as 5 weeks after the birth of the mice, and this mineralization progressively gets worse, more involved, so it's a progressive biomarker of the entire PXE process in these mice. And this just shows the mineralization. These are special stains which pick up calcium, all here. Another stain, we see this mineralization. If you look closer at this area, there's connective tissue and there's some calcium deposits right there. But then if you look later in the mice, you look other tissues. This is a part of the leg. Here is a blood vessel, and if you look closely into the blood vessel, there is clearly a mineralization taking place, which tends to occlude the blood vessel so that blood cannot go through very readily here. And this is from the back of the eye. Here's the retina, which is the layer that contains the photoreceptors. In other words, the vision is concentrated here. And out of this retina grows up these kind of mineralized areas, which then cause the breakage of the membrane, bleeding, and loss of vision. So all these take place in the mice. Here you see this causes the breakage in the back of the eye. And again, in the skin, similar to human skin, you see this mineralization. In other words, deposits of calcium and phosphate in the skin. Now, so PXE mouse, in other words, it has the same gene alterations as in PXE patients. So it's the lack of the functional protein that serves as a pump in the liver. And in fact, these mice, they have the same genetics, they have the same kind of histopathology and ultrastructural features as PXE in humans. So we can now use these mice as a model to study PXE. And as I pointed out, this is important to understand. In normal mice, we haven't manipulated these mice at all. Up to 6 months, there's clearly no calcification of the vibrissae at all. But if you have the those mice where we eliminated the APC6 gene, at 6 weeks, there is clearly mineralization starting to take place. 6 months, there is more of it. And 22-month-old mice, we see lots of mineralization, so their disease is getting worse. So why study PXE in mice? Why not in humans? Well, one is that humans are all very different. Humans have different genetic backgrounds because we all are a mixture of things from the past and present. While mice, you can breed them so that they all have very genetic background. They look the same, and the only difference in these mice is that they either have or they don't have expression of this gene. The second point is that mice have relatively short lifespan. Their total lifespan is 2 to 3 years, while humans is about 70 to 80 years. And for example, puberty, the mice reach puberty about 5 to 6 weeks, humans about 13 to 16 years, so it's much faster to study a disease obviously in these animals than in humans. Also, the mice are all in a uniform diet. They have the same bland chow, but we know exactly what is in it. All the mice get the same diet, so you can manipulate the diet, add some components, take something off. And you can breed lots of mice, so you can study lots of mice at the same time. You can study the effects of diet. You can test various kind of drugs. You can test various treatments, such as gene therapy treatment, which all are going on now in different laboratories around the world, including ours here in Philadelphia. So let me just illustrate the use of these mice for studies on PXE, and specifically studying the effect of diet. These are the studies by Jennifer LaRusso, who's in my laboratory here in Philadelphia. She's actually a dermatology resident who has been working several years in the lab and now also learning the clinical dermatology, so she's going to be a very well trained dermatologist with knowledge of PXE. And she has specifically looked into the effect of magnesium on a PXE. And originally, we already published some of these studies. Originally, we had a preliminary study where we looked into these mice, the effect of various components, specific mineral modifications in particular. And what we measured was this calcification of the vibrissae, the whiskers, as a sort of indicator of the mineralization process. And one of the goals of this early study was exactly to examine the effect of phosphate. If we increase the phosphate content of the diet in the mice, does it make the disease worse or perhaps better? And this was based on the early reports, go back to about 20 years, which suggested that people who are eating lots of dairy products, especially when they are young or during adolescence, if they have lots of dairy products which is high in phosphate and high in calcium, that their disease, PXE, might get worse. So that was the starting point, and we then made a custom designed diet for the mice. And as compared to the normal mouse diet, we actually increased the phosphate content twofold. There's about 0.43% to 0.85% of the total content of the chow. And there was also actually a decrease in calcium and a decrease on magnesium. And then we looked into the vibrissae of these mice and measured if their mineralization got worse or better. This just shows the kind of technology you use computerized. You see these darkened areas, that's all where the calcium is. This is the capsule surrounding the whisker, and the computer can calculate what percent of the total area is mineralized. So we can get specific numbers, and we can compare those two different diets at different stages. To make a long story short, in this case, our phosphate and calcium changes actually caused the PXE to get worse in mice as compared to knockout mice here on normal diet. These experimental diet mice, their mineralization got worse. But this was sort of a preliminary study, and it was not very clear if the worsening of the disease was secondary to increase in phosphate, or did it have something to do with decrease in magnesium, or combination of both. And also there was change in calcium, so it was not very clear which of these components of the diet or minerals really played role in worsening of the disease. So we made a more definitive study, which actually was just published earlier this year, and it basically demonstrated that if we increase the magnesium content in the diet, that actually prevents the mineralization or the calcification of connective tissue in this mouse model of PXE. Now, whether it does that in the humans, we don't know, but certainly did in the mice. And I can show here, this was what we used, different groups of diet. And this is how much we had phosphate and how much we had magnesium in the control group. And the key group here is the group C, so-called high magnesium group, where they had the same amount of phosphorus or phosphate as the controls, but we increased their magnesium from 0.22% to 1.10%. In other words, it's a five-fold increase in magnesium. And we placed those mice on this diet at 4 weeks. That's at the time they wean from the mother's milk, and they were then placed on this diet, 5 mice per group, so you can study controlled amount of mice. And we then compared the mineralization in the control group and the high magnesium group. And this just shows an example. If you look 8 weeks later, here is those mice that have been on controlled diet, and you see those darkened areas, they have lots of mineralization, lots of calcium deposits in their tissues. While those mice that have been in high magnesium diet, there's absolutely no evidence of mineralization at all. So therefore, just increasing the magnesium, everything else was the same. The mice were the same, they were litter mates, their diet was the same, except they had 5 times more magnesium in their diet, and they did not develop PXE. And we can quantitate that again by various means, but this shows here's a wild-type normal. This is our knockout mouse. And if we now put those knockout mouse to a high magnesium diet, they don't develop mineralization at all, so they are like the wild-type mice right here. Now, this clearly suggested then that the development of PXE can be prevented or counteracted by high magnesium diet. And again, I have to stress that this is in the animal model of ABCC 6, the PXE mice. We do not know the specific role of calcium or phosphate, and certainly the role of magnesium requires further investigation. And again, in order to translate this to the patient level, we really have to have controlled studies. We have to be aware of the potential side effects of this manipulation, and those kind of studies will probably be coming in the near future.
Dr. Uitto, I have a couple questions.
Yeah, please. Actually, let me just finish, because this is, I think, is right at the end. So, basically, this is just an example of one of the studies that is going on. We have lots of perhaps more demanding, more sophisticated studies, such as gene therapy studies going on, trying to put the gene back to the liver of these mice, trying to identify the molecules that are causing the mineralization. But this is just an example how these mouse models have been extremely useful for studying PXE, and I'm sure they will be even more of utility when we start testing different kind of drugs, different diets in detail, etc. So just to finish up, yes, this is actually, I'm right here, see no beard this time. These are people in our laboratories who work various kind of diseases, not only in PXE, but various other heritable systemic and skin diseases. And this is just to acknowledge the people who have been instrumental for our studies. I have two very talented young scientists in the laboratory. Jia Jiang is in charge of the mouse work, and these are all people who have been helping her. Here's Jennifer LaRusso, who did those magnesium studies, and all these other people are involved in different other projects. And then we'll be looking into the genetics of PXE, trying to understand the modifying factors, other genes, etc. And these studies are led by Lily Lee, who also has been helped by a number of people. And I just want to thank at this point our collaborators and advisors, our Hungarian colleague András Varadi, who was visiting our laboratory under the sponsorship of PXE International last year. And we visited his laboratory just a couple of weeks ago, and we have plans to actually find out what is causing PXE, exactly how we can interfere with it. Joel Rosenblum is a connective tissue expert here in Philadelphia, and I want to thank Sharon Terry and the entire PXE international community for supporting our studies, and obviously we are very pleased of this collaboration with the patient advocacy organizations. So this is just an example of what's going on in the PXE research, particularly in the studies that use animal models, particularly mouse model that clearly is mimicking the PXE. So, Chris, this is what I had to say at this point. Hopefully there was some interest, and you said there were a couple of questions.
Yes, we do have a couple of questions. The first one is about, well, I'll just read it to you. I wonder if the mouse studies are comparable to human twin studies, because the mice are on a controlled diet and in a controlled environment. And since the PXE population is so limited and twin studies are not likely, twin studies look at how the environment and diet affect genetic diseases. Could the mouse studies be interpreted as like a twin study to determine the role?
Absolutely, that's a correct statement. Specifically, we are talking of identical twins, and identical twins have pretty much the same genomic makeup. In other words, their genes are very similar. And the point of these mouse studies, indeed, is that we have bred these mice so that their genetic background is very similar, if not identical, to each other, and to adding to the utility or usefulness of the mouse model is that we can actually feed them exactly the same diet. I'm not sure identical twins want to eat the same food all the time, but yes, in principle, these are very similar to the identical twin studies which, as the question appropriately pointed out, there are not too many identical twins with this relatively rare condition.
Thank you. There's another question. Are any mice studies being done to attempt to reverse the mineralization in adult PXE mice?
Yeah, that's our mineral the diet studies. Actually, they were designed initially just to start at the time when the PXE in mice starts. So in other words, we were really looking for prevention of the development of the PXE in the mice. Now, we have started, we have not completed the studies where you actually try to see if you can reverse the mineralization. And in those studies, you start changing their diet later, about 3 months of age. And we are right now having those mice coming. They are actually soon to be at that age. The problem with these mouse studies is that you have to wait for the first 3 months, and then in order to see an effect on the reversal, unless it's a very rapid reversal, it's going to take probably several months, if not up to a year. So yes, those studies are in the planning stages, and obviously it's kind of important follow-up to the prevention studies. And I'll certainly keep you all posted once we have the results coming.
Thank you. We have heard that the gene expresses in the liver and the kidney. You spoke mostly about the liver, and that's what we mostly hear. What do we know about its expression in the kidney?
Well, I'm mostly talking of the ABCC6 expression in the liver, because the expression in the liver is clearly the highest. It's much higher than in any other tissue. It is also, as I mentioned, also expressed in the kidneys, but the level of expression is not as high as in the liver. And then if you look at the tissues that are really affected in PXE, like skin, or back of the eye, or the blood vessels, their ABCC6 is not expressed much at all. Well, it's not very clear, or it's not definitively determined what role kidney expression might play. And in fact, we had plans for a definitive experiment where we would do liver or kidney transplants to the mice. In other words, you replace the deficient liver with a liver from a healthy mouse. Those are very technically difficult because the mice are so small, and seems to be there is nobody in the United States who can do mouse liver transplant readily, but that would be an experiment that actually we have been contemplating, haven't done it. So to make the answer clear, we do not exactly know what role of ABCC expression in the kidney does have.
Thank you for that. There were some questions that were sent in before the webinar, and you have a copy of those. I'll read them out if that's okay.
Yeah.
You have an article called Mutations in the GGCX and ABCC6 genes in a family with PSEUDOXANTHOMA elasticum-like phenotypes. Can you tell us what the role of the mutated GCCX gene plays in PXE? And do PXEers have one? And do they need to have one or both of those mutations to have PXE?
Yeah, that's a very astute follower of the research there, because our publication just came out probably less than 2 months ago, if I remember correctly. And basically, as I explained, and you all know, PXE is caused by mutations in the ABCC6 gene. And we now know that PXE is a recessive disease, autosomal recessive condition. That means that the affected individual has to have mutations in both copies of the gene. In other words, one copy from the mother and the other copy from the father. They both have to have mutations, because just one mutation in one copy does not cause the disease. These individuals are carriers of the mutated gene. Now, we ran across a couple of families where we found only one or none or neither ABCC6 allele had mutations. And instead, they had a mutation in this so-called GGCX gene. It's a completely different gene, does completely different job, and these are very rare families. In fact, they're probably less than about a dozen in the world's literature. We looked into a couple of them, and one of their features, especially if they have mutations in both copies of the GGCX, that they get PXE, but they also get bleeding tendency. They get so-called vitamin K dependent coagulation deficiency, which is a rare condition in itself. Now, why we reported this? We reported it because it has been giving us insight as to what is going on in PXE. And there are hypotheses that relate to vitamin K status in patients with PXE, and again, those are under investigation. We don't exactly know what vitamin K's role might be, and as I said, these GGCX gene mutations are sort of exception, but very kind of helpful exceptions to understand the disease. But the majority, probably 98% or 99% of the individuals with PXE have mutations in the ABCC6 gene. I hope it makes it clear.
Thank you. There's another question from this attendee. Are there any other genes that play a role or are suspected of playing a role in PXE?
Yes, so the primary mutations are, as I said, in 99% of cases in ABCC6. There are these rare families where the primary mutations is in GGCX gene, and there is no other gene that we know of that would be the primary cause of PXE. Now, as you all know, PXE is highly variable in its severity, and there has been some thoughts of modification of the primary defect. It looks like diet is certainly one factor that modifies, but also it's very clear that people's genetic background, their inherited background, different kind of complements of genes that can modify the presentation, the severity of a PXE. And that theory, in fact, is not theory, at least in mice. Again, we use the mice, and we have a paper coming out probably in a month's time, which demonstrates that you can modify the severity and the time of onset of PXE in mice by just changing their genetic background slightly. So there are modifier genes. There are a couple of suspected modifier genes in humans, and again, there is work going on to identify the nature of those modifier genes and see if you can somehow interfere with their expression so as to minimize the effects of PXE, although not completely eliminating it in the affected individuals.
And the last question there is, has there been any research on epigenetic effects and PXE, and if so, what are the results and the findings?
Yeah, epigenetics. Epigenetics is the current vogue to look for genetic factors that change the gene expression of the primary gene. And that sort of relates to the genetic background modulation. No, there is no, to my knowledge, any specific work on epigenetic effects and PXE, although that certainly should be a very useful area to study, a similar way as these modifier genes. So there are factors that can change the manifestations, can change the time of onset, and can change the severity of the disease.
Thank you. And there's a question about magnesium. Why did you choose to look at magnesium? What made you suspect that might improve the mineralization picture? And is there any evidence that magnesium might be toxic in humans in large doses?
Yeah, good points. Very good points. One is it was a little bit serendipitous in that when we did the preliminary study with minerals, we were concentrating on our focus on phosphate and calcium because of the historical reports, as I said, going back to probably 1980s, suggested that high phosphate and high calcium intake makes PXE worse. Now, it just happened that the diet we had, it was a commercial diet, happened to have a low magnesium. And those mice got worse. They got worse PXE. So we weren't sure whether it was the high phosphate or changing calcium or low magnesium. And when we went to literature, there's clearly quite a lot of literature on the effect of magnesium on bone development and mineralization of the bone. And it all suggested that the mineral, specifically magnesium intake, can change the mineralization in the bone. So we wondered, can it change the mineralization of the soft tissues in the skin, the eyes, the blood vessels? And therefore, we designed this new study to increase the magnesium to 5. Now, there are at these concentrations in humans, there is not very good literature about side effects. And the one concern that needs to be tested, and we are testing now in mice for long-term use of magnesium, whether they also prevent somehow mineralization in the bones. In other words, you cure PXE, but then you get osteoporosis and you break your bones. Now, that's not necessarily a very good trade-off. So that is one of the areas that needs to be looked, and that brings up to the whole issue with any of these treatments, drugs, and modifications in the diet, etc. What are the risk-benefits ratios? You certainly have to be aware of the side effects and then balance them with the potential benefits. So all this, specifically with regard to magnesium, that's all going on, and the only thing I can say that it clearly worked in mice. We do not know in humans. I do not necessarily recommend people start taking magnesium until we have controlled studies and know all its risks and side effects.
Thank you for that. We have another question. If a treatment such as stem cells was developed to treat vision loss, what would prevent the problem from recurring if the ABCC6 defect is still present?
Yes. In fact, we are working on stem cell research. And what we specifically try to do is put stem cells to the liver. And those stem cells would, as you know, stem cells have a potential to differentiate to different organs. So you start from the very undifferentiated cell, you put them into the right contexts, like into the liver, and they develop to be new liver, basically, new liver cells. And so it replaces the existing liver. And if the change is permanent, then the ABCC6 deficiency will be replaced by normal liver tissue. Now, in a very simplistic way, you could probably reach the same endpoint by doing liver transplant. And then again comes the issue, is liver transplant with all its risks and complications, is it worse than, for example, having the mineralization problem in the skin? All those kind of things need to be worked out first in the preclinical animal models, as we are trying to work in the mice now, and then subsequently through carefully controlled clinical trials.
Thank you very much. I think that's it for our questions. I'd like to thank you very much for preparing this presentation for us and being here with us today. It's been excellent to be able to ask you all these questions. And I would like to, Dr. Uitto, do you have the last slide that shows the next webinars?
That's absolutely here.
Oh,
so let me let me just thank PXE International for inviting me and having here, and I hope our studies will lead to development of treatment and perhaps eventually cure for this condition.
We are very hopeful for that too, and we watch your research very closely. I wanted to remind everybody that we actually have a lay summary of the research you just described on our website in the research section. And we don't have the research section showing on this slide, but you can find it pretty easily if you go to our website, www.pxe.org. And this webinar was recorded, and you will all be able to hear it and watch it again, or anyone who hasn't seen it can watch it from our website in the near future. And we don't have any webinars right now planned until the new year, so we don't have any to tell you about, but the webinar registration is from the website as well. I think another question came in. Would you mind answering another question?
No problem.
A follow-up to the stem cell question, would a retina stem cell treatment also need a liver stem cell treatment?
No, that's a little complex situation. The primary, how we see it, the primary side is obviously in the liver, and then you, as a result, you get calcification in retina, and that basically causes problems with the blood vessels and bleeding, etc. Now, if you do stem cell transfer directly to the retina, even if it changes, yes, that's a good point. So if you do it the retinal stem cell transfer, that may give you a temporary improvement, but the person who asked the question is absolutely right. It does not correct the underlying primary metabolic defect, the one that is caused by deficiency in the liver by the absence of ABCC6, and it would be quite conceivable that the eye problems may get temporarily better, but then on the long run it's not a cure.
Okay, well, thank you. We have a slide now of the research section of the website, so I'm going to show that by changing the presenter, Dr. Uitto. So, and I'd like to thank you. Okay, here we go. I'd like to thank you again for being with us. We have updated research news on our website at this research link right here, and the past webinars will be on the website for anyone that missed part of this due to a time zone difference. And thank you very much for being here with us, Dr. Uitto.
It's been my pleasure. Thank you.